Yeah, good morning everyone. Welcome to the 9th session of Current Affairs, Current Edge sessions. So today we are going to discuss healthcare and biotechnology related developments. Let's get started. First, have a broad overview on what healthcare has got in recent budget. Tell me what you know about in this sector. 1.5% of 1.5%. 2.18. Any other suggestions? Healthcare got some 2.1% in 2022-23. That financial year is ending here. This year it got slightly more than this. So 2.18 something is more fair decent number, not 1.4. 1.4 was earlier number in 2019-20. Post COVID times we have considerably increased this health expenditure. Now what is this target? National Health Policy 2017 target. What is it? At least 2.5% was the target. That can be met if this trend is followed further. What else? There were major, what was the most major schemes? Ayushman Bharat scheme, Pradhan Mantri Jan Arogya Yojana. What are specific features of this scheme? It is providing primary health care or not? It is providing secondary and tertiary healthcare services only. That is like a insurance scheme, having a coverage of 5 lakh per year per family. There is no limits on number of people per family or any other criteria is not there. Every family is applicable for with this insurance bracket. Now this is world's largest public funded scheme having a target beneficiary of 50 crores. Go ahead. Proposed elimination of anemia by 2047 and sickle cell anemia elimination is also proposed in this budget. Now if you look at the budget expenditure for last few years, the graph is not straight line. If any statement is there health expenditure is continuously increasing or continuously decreasing, that is not correct. It is having 65,000 allocation during pandemic times. It has been reduced to 58,000 this year now. Last year it was more reduced to 55,000 crores. Okay. Only trend is important, the numbers are not important. Trend is having some zigzag trend, at least know this much. Go ahead. Now what is anemia? What is anemia? Tell me first. Shortage of red blood cell in body. What red blood cell is doing? What is the function of red blood cell? It contains hemoglobin. The function is carrying oxygen to different parts of the body as well as carrying carbon dioxide from different parts of the body to lungs. Carrying these gases is the main function of RBC. This is done by hemoglobin. And which pigment, which exact element is doing this? Iron or iron. Now anemia is more associated with deficiency of iron. National Family Health Survey 5 says that more than 52% of women in India in the reproductive age are anemic. More than 52% means that is considerably huge number, extremely huge number. Now why this anemia is having? What are the causes of anemia? Diet issues is there, loss of blood during menstruation for women, worms in infection can also lead to this anemia situation. Other diseases like kidney issues can also lead to, or body's inability to absorb iron. Different conditions can lead to anemic situation. Anemic is a situation that different ways it can get into. And what is the treatment? Mostly diet, having proper balanced diet is the treatment procedure. And what is one major step government has taken in this regard in this year? That is that. Government has started distributing Fortified rice. What is fortified rice? We'll see in the next slide. We'll have in detail. So this is a broad picture on anemia. What next? Sickle cell anemia. What is sickle cell anemia? Anemia means lack of normal anemia. Lack of hemoglobin is the due to lack of hemoglobin. These gases, oxygen and carbon dioxide cannot be transported within the body efficiently. Here it is not, problem is not lack of red blood cell or lack of hemoglobin is not the issue. Here issue is normally red blood cell have round shape. Due to some XYZ reason, the blood cells are having different shape. Now different shape means there is two issues. One, it cannot transmit like the other normal blood cell, red blood cell is doing. Second, this shape, due to its shape, it can get blocked in some XYZ parts of the body. And this block will also lead to creating pain and other issues. Now this is called sickle cell anemia. Why this is coming? Why? What is the reason? Mostly it is a genetically inherited disease. And where is the issue lying? Issue is lying where blood cells are generated. Where blood cells are generated? In the bone marrow. So genetic disorder is the major cause for sickle cell anemia. It is some issues related to bone marrow is the major reasons. Now tell me, symptoms you can read from these infections, prone to infection, severe pain will be there, delayed growth, vision problem, anemic issues will be there, all these will be symptoms of the disease. Lifestyle is the solution XYZ. Now what is the major treatment options for this sickle cell anemia? Bone marrow or stem cell transplantation. That is the only permanent solution available for sickle cell anemia. This also mostly it is only conducted in children only at very young ages, and that too there should be a donor which have very similarity with the children. Mostly his siblings bone marrow will be adopted. If he grown up, then changing the bone marrow, that is very complicated. It is mostly not possible. So this is the only treatment that is available to tackle the sickle cell anemia. Go ahead. Rice fortification, what it is? Don't confuse it with genetically modified rice or any XYZ big technologies. Rice fortification means you have this normal rice, 10 kg of rice, you add a modified form of kernels. Kernels means this small rice. You have artificial kernels added into this. And these artificial kernels will be small in quantity and this will have iron and other nutrients. Okay. That is fortified rice. 1:100 ratio will be used. What else? Different phases it will be implemented, covering ICDS, PM portion, that will be primary objective, first target. Then it will be expanded to TPDS. What is TPDS? First it will be implemented for children and portion abhiyan, like lactating mothers, pregnant women and all. Then it will be expanded to PDS systems in phased manner. Go ahead. This is different process that is happening. Rice flour and premixed protein, all these premixed nutrients, all these are mixed together to form this whatever that is fortified rice. Is this rice having higher storage time or lower storage time? Like lifetime, how much you can store? Fortified rice have longer or shorter? Shorter. It is having at least 3 years plus of this shelf life. It is not that much short also. Cannot call this as shorter because so many processing happening, rice quality is also improving. Go ahead. What is Indian Biological Data Center? This has been recently launched by Minister of State for Science and Technology. It is a dedicated data center for biological applications. all life sciences data generated from publicly funded research in India. This data will be XYZ saved in this data center. It has a capacity of 4 petabytes. Where it is? Faridabad. Regional Center of Biotechnology, Faridabad, that is, it is located, having a disaster recovery site at NIC Bhubaneswar. Where is Bhubaneswar? Odisha. Go ahead, very factual content. Now let's look at some diseases in news. This is also very factual thing. You just have to understand, these are bacteria, examples of bacterial diseases. Tuberculosis caused by Bacillus microbacterium, spread through air, like infected person if cough or something, then it can spread to the other person through the air. Mostly affect lungs, but it can affect other parts of the body also. Anti-TB rifampicin, that is the drug used for tuberculosis. Leprosy, what is leprosy? It's also bacterial disease caused by Mycobacterium leprae. It is also transmitted through cough or sneeze droplets. Skin peripheral nerves, it is infecting. The major problem is after symptoms only started generating after 3 to 5 years. By that time it have created a big issue and treating will become very tough. There is BCG treatment for this. What else? Salmonella typhimurium is this virus bacteria that causes this from eating raw or uncooked food. This bacteria, where it lives? In the intestines of different animals. Now if you eat this animal without proper cooking, without doing something like China's people, all this will get infected of this more, because they have this practice of eating animals alive, eating without partial cooking and all. So this is such kind of a virus bacteria. Now we have Shigella bacteria. Shigella bacteria is causing this. It is transmitted through contaminated water, contaminated food. Person to person contact also lead to transmission of this. There is no vaccine, there is no exact treatment for these diseases. Treatment you can take for reducing the symptoms. Treatment you can take for not exaggerating whatever sickness you are having. But exact treatment for these are not evolved. Go ahead. Very factual content it is. AIDS acquired immunodeficiency syndrome, it is caused by human immunodeficiency virus, HIV virus. Through body fluids it will be transmitted, like through blood, breast milk of mother to baby, sexual contact, all these lead to transmission of this. Immune system will be affected. Antiretroviral therapy, ART, will be treatment for dengue. What causes dengue? These all are viral diseases. Flavivirus, it is spread through bite of Aedes mosquito. It affects liver. No specific treatment is there. You have to take proper food, normal medicines for fever and all this. Zika virus, it is caused by the same virus itself. It is also spread by Aedes mosquitoes biting. It is also spread by sexual contact. Now headache, rashes, malaise is the treatment. No symptoms. There is exactly no treatment or medicine for Zika virus also. What else? Japanese encephalitis, bite of Culex mosquito, different kind of mosquito is leading to spread of this. Brain will be infected or affected region of this. Then dangerous for children. If children are infected, it is more dangerous. There is no specific treatment for this also. Now what is measles? Measles is caused by paramyxovirus family. It is passed through direct contact or through the air from cough, sneeze and all. The infect is respiratory tract. No treatment here also. No treatment means doesn't mean you don't have to take the treatment. No specific vaccine or no specific dedicated treatment medicine is there for exact diseases. Go ahead. If factual info, this is protozoa diseases, malaria and Kala Azar. Kala Azar is quite important for two reasons. Why? Government has come up with a statement, we will eliminate Kala Azar by 2024. So if government minister is saying something, then there is high chance. Secondly, there has been some vaccines developed for Kala Azar. Some vaccine development is happening. This was also in news. So for these two reasons, Kala Azar is quite important for this exam. Malaria, it is caused by Plasmodium. Bite of mosquitoes is leading to spread of this. Affects RBC and liver. Mosquirix is the vaccine for malaria. Similarly, Kala Azar is caused by Leishmania donovani. Female sandfly it is spread by female sandfly. It affects bone marrow and liver. This is the new vaccine that is developed for Kala Azar. Go ahead. Nothing much conceptual here. You have to read this and to somehow memorize this. At least you should be having a knowledge of this is bacterial disease, this is viral disease, this is protozoa based disease. Go ahead. These are some new developments in vaccine technology. What is Siravavac? This is India's first indigenous developed quadrivalent papillovirus QHPV virus vaccine. Where it is used? Applied for cervical cancer. This is the only form of cancer that has vaccine available. Developed by Serum Institute of India, Pune. What next? Incovac. What is specialty of Incovac? Again, India's first intranasal vaccine. What does it mean? It would be taken through nose, not injected into the body. It is developed by Bharat Biotech. It has been approved for primary immunization. Primary immunization means? In those primary means in those people who doesn't took any other immunity vaccines or any other immunity XYZ. Yes, infants also. Lumpiprovac, where it is used, it is not a human vaccine, it is a livestock vaccine. It is used in animals to prevent lumpy skin disease. You might have seen these cows with small bubbles in their body. So to prevent this XYZ vaccine is being implemented. Go ahead. One Health, what is One Health? Yes, it's a holistic approach to it. We are not putting anthropocentric world approach. Anthropocentric world means it have a views that human is the most important or most evolved species and priorities are humans are the most important. That is anthropocentric view. We are going ahead from this. We are taking environmental perspective, human health perspective and animal health perspective. All are quite important. And integration of this is called One Health. Why is it in use? One Health helps in all these examples, all these benefits. Some question is there, what is XYZ benefits of One Health? You should be identify it with potential threat from human-animal interface. What is the best example for this? Even if COVID come from bats, then it also came from due to these human-animal issues. Nipah virus, all these zoonotic viruses, that is coming due to this human-animal interactions. Second, antimicrobial resistance. How One Health helps tackling AMR? AMR is different concept now, not vulture concept. AMR is antimicrobial resistance. What it is? Not overdose. Not overdose also. Like TB is there. You have first line of drugs. Now if you don't take this properly, if you miss this, or if your TB bacterium has acquired characteristics to fight with this drug, then what will happen? All these drugs, even if you take, it is not going to impact. Then you have to take second line of drugs. Second line of drugs, this itself will be having some 7 tablets per day. This will be having like 50 tablets per day. That much high intensity medicines you have to take to tackle this bacteria then. So bacteria acquiring more characteristics to defend the first set of drugs. That is called antimicrobial resistance. Why this is happening? Either you will be taking these drugs improperly, so that you will giving bacteria enough time to adapt and evolve new characteristics, or it can also not only just human bacteria or human aspect, poultry farming, meat sections, all these animals where it is used. If there also these drugs are used without any control, without any limit, that can also lead to this all aspects. So if you approach this One Health in principles, animal health will also be upholded. So antimicrobial resistance can be reduced, ensure food safety, prevent environment related health threats. All this you can associate with, protect biodiversity. These are all benefits of One Health. If some question comes, then you should be able to identify. Then recently a joint strategic framework for One Health has been published. This has been published by many organizations. FAO is there, World Organization for Animal Health is there, World Health Organization is there, UNICEF is also there, and World Bank is also there. Like many organizations came together to publish this framework. India has also started a Center for One Health. Where it is? It is at Nagpur. And India has also started a national expert group on One Health. So due to some action by government, international organization, this is somewhere in the news. At least you should know the concept. You should know if some statement come from here. Go ahead. What is the next topic? Antimicrobial resistance. This we have been talking about. Same concept what I have discussed so far is written here. It can come through animals, it can come through humans misusing these drugs, can come from different sources. Even it can come from plants also. Okay. Concept is same, rest of the things you can read. Go ahead. What is this? This is a new technology. Chimeric antigen receptor T. CAR T cell therapy. This was in prelims. Okay. What is it? What is it technically? For what treatment it is used? CAR technology. Treatment of cancer. How it is treating cancer? Tell me. What is cancer technically? Uncontrolled growth of XYZ organs. Issues will be mostly with WBC. Now what this does, you take this is the patient, you collect the blood from him. Now remove the T cells. T cells are a part of WBC. Now you remove the T cells from these patients. Now what you do with T cells, you do some genetic editing on this, genetic modification on his T cells. Now you had extra features to it. Now what these extra features will do? If you see a cancer or tumorous molecule, it will go and attack this. What is the function of this white blood cell? If any foreign particle is there, then go and attack and kill this. If any new virus come into my body, this white blood cell T cells will be going there and attacking. So we are giving some genetic modification. T cells are giving some extra tools so that it can go and kill cancerous virus. Then the patient will be subjected to chemotherapy and this T cells will be put into his body. Now this new T cells will go and attack the cancerous cell and he will have the relief. That is the whole concepts of CART technology. It is for cancer treatment. WBC is the altered to treat the cancer. T cell is a part of WBC. Yes. Chemotherapy is normal treatment for cancer. That is not something new as a part of this. That is on the process, that way it is shown. Ideally, it is taking the T cells from his body, genetically modifying it, then reintroducing it into the body so that it can attack whatever cancer or tumorous cell is there. Go ahead. These are some examples. So we have discussed this in other class, current affairs class also. Red blood cell, RBC, red blood cell. What is it for? These are specification. Non-nucleus contains hemoglobin. That is why it is red in color. It is having this much of 6.5 to 8.5 micrometer diameter. Transporting oxygen and carbon dioxide is the main function. These all are different types of WBCs. Their functions and their descriptions and functions are given here. Mainly what is the function of WBC? Immunity is the major function. Any foreign body came, how it attacks? Yes, different methodologies there. It is increasing temperature, or it can directly engulf this material and eat it, or it can produce antigens. Antigens have specific proteins. These proteins go and attack these antibodies. So there are different mechanisms and different names for different mechanisms. Basic function is giving immunity to the body by attacking whatever foreign particle it is. Now there is one more thing. What is that? Is platelets in the blood. What is platelets? Plasma membrane of the blood. What is the main function of platelets in the blood? If any cut happen, after some blood is gone, there clotting should happen. This clotting happens because why? Because of these platelets, in the presence of platelets in the blood. Somebody XYZ don't have these platelets for some reason, then some come happen, the blood will continuously go on for longer time. Go ahead. What is gene and what is genome? Tell me what is the difference? Gene is particular. Gene is a particular segment, particular portion of DNA of a molecule that is called gene. If you take inside a molecule, there are DNA lines. If you take the DNA, that is called gene. Whereas genome means, if a human body is there, the whole human body, everyone will having same genome structure. Every human will be having same genome structure, but gene in everyone will be different. Every person will be different. That's why everyone is having new XYZ characteristics. Genes code for proteins. Gene itself cannot code for proteins. Genome means like whole DNA ka the arrangement structure name is genome. What is function of this gene or DNA? We'll see, I think, next slides we have. It consists of few base pairs, as it represents only a DNA segment, consists of 4 base pairs in the cell. What does it mean? We have DNA as well as RNA. DNA is having different kind of base pairs, RNA is having different kind of, some difference is there, 2 base pairs are different and all. So this will be having only those which DNA has, while this having this completely list of XYZ arrangement of every pairs. That is called genome. This is called genomics, it is called genetics. Individual has thousands of genes in him. An organism has one genome, that is human has only one genome. That is Human Genome Project means you identify the order of DNA or order of bases arranged in human XYZ. That will be similar for all. Go ahead. Cell structure, give me some information on this. There has been question asked from this. Cell what all contains? Nucleus. Nucleus, what is the function? It is like control center of the cell. It has DNA information. What is the purpose of DNA information? Transmitting genetic to the next generation. If a cell is multiplying, then new cells will have characteristics of first cell because there is DNA. Doing this hereditary transmission of characteristics, that is a function of DNA, which is mostly in the nucleus of the cell. Are DNA only in the nucleus of the cell? Where else you can find this? In mitochondria also you can find some part of the DNA. It is said that around 99.75% of the nuclear DNA is in nucleus. Very small portion, that is 0.25% is in mitochondria. What is the main function of mitochondria? It is the powerhouse, kitchen of the cell. It produces energy, whatever is required for all these functions. Then you have major organ, or this you can see which are different organs. Vacuole is there, other plasma membrane, other membranes are there. Golgi apparatus that modify the proteins, lysosome that digest the food materials and waste products handling. All these are different organs of cell itself. Now what is the major difference between plant cell and animal cell? Plant cell are much larger. Yes, you are right. Plant cell consists of plasma membrane and cell membrane, all are part of plant cell. Animal doesn't have all this. Plasmodata is present. What is this? Discussed in last class also. What is this plasmodesmata? Too many plant cells are there. There will be some connection in between. So the material as well as information will be flowing from one cell to another in plants. This is only present in this connection will be there in plants only. In animals, most of the cells are independent. There will not be any direct connection pipes between these cells. That is present only in plants. Now vacuole, what is vacuole? There will be a large hole within the cell. In plant cell, there will be one big hole almost in the center. Whereas in animal, there will be small micro holes which even you cannot see. Chloroplast means chlorophyll element is there. Tonoplast means some kind of outer membrane of this vacuole. Vacuole is bigger in plants, so it is more provident in plants only. Rest you can see lysosomes are very rare in plant cell while it is present in animal cell. Storage strength is starch grain. Storage is glycogen granules. Question mostly comes on basics only, so just understand the XYZ basics and leave it. Don't over research. What is prokaryotic cells and eukaryotic cells? Tell me the major difference. Prokaryotic cells, the cell is not having different segregation like this. Here there will be a vacuole, there will be a nucleus, there will be mitochondria, everything will have its own cell wall, and inside this it will be there. Here like everything will be there but distributed here and there. There will not be segregation for all these parts of this. The rest of this you can read from here. Can these prokaryotic cells have DNA? It can have DNA, it can have RNA, it can have everything. But it cannot have differentiation like this. Prokaryotic can have this virus, many viruses are having prokaryotic structure. Go ahead, rest, I think you can read this. Very factual information is there. Go ahead. What is major functional difference between DNA and RNA? DNA, as I already told, what is main function of DNA? To transmit the hereditary XYZ. So hereditary characteristic storing is the function of DNA. And what all includes this hereditary characteristic? Skin color. If human we take skin color, all this skin color everyone's is inherited itself. Height of the body, different these things are all inherited. So storing this is the skin color. This part is done in DNA. Now suppose where is the skin made? Skin will be generated in some other parts. Information is stored in DNA. Now skin production will be happening in some other part. Now there should be a mechanism where you take out this information, then give the XYZ to skin production part. Make like this. This information transmission, who does this? That is the function of RNA. This is a double helix structure is for DNA. Now when you need to transmit the information, what happens? DNA splits into this like this, single helix structures. Now these single helix structures carry the messages for whichever organ or genetic messengers for production of different organs. This they will be taken into that organs, and that organ will produce amino acids. Then amino acids will transferred into proteins, and proteins will become skin, and all this process will happen. So data transmission is the function of RNA, and data storage is the function of DNA. That is the fundamental difference. Rest you can read from here. It has 4 bases: adenine, guanine, thymine and cytosine. It also has 4 bases but different. Uracil, guanine, adenine and cytosine, one is different. Location: DNA is found in nucleus as well as mitochondria. whereas RNA is formed in cytoplasm, nucleus and ribosome consist of RNA. Sugar portion it has 2, it has ribose. Function we already discussed. Predominant structure is double helix, here it is single helix. We solved one PYQ question where what was that, something like a DNA was given and DNA was having single helix structure, something like that was given. So if you at least know the basics, RNA given in the question having double helix structure. So at least in the basics you know 21 question. You know that RNA is mostly not having double helix structure. So unless you know there is any specific RNA having this structure, if you know that knowledge, then you take it as right statement. Otherwise that is against our basic fundamental knowledge. So in biology, lot of questions we solve like this. Questions, even though seems very tough, but if you have the conceptual clarity of what is the function of X, Y, Z, basic things you know, you can apply that and solve to some extent. There was a question on RNAi. What is this? RNA interference technology. Statement seems absolutely difficult questions, but at least you know what RNA is doing, what can XYZ happen. Basics of RNA you know how to apply to some level. You can reach answer for this question. We solved it in PYQ sessions also. So that's why we are doing all this. This is not purely current affairs stuff, but in biology so many questions you need absolute clarity on these aspects. Okay, these are theoretical also. Go through this all what is written in these notes. These notes will also be provided to you. Don't take too much stress on writing down also. These all will be provided. So take down these notes, revise this. Have that enough conceptual clarity. Go ahead. What is stem cells? What is stem cells? What is their function? It can transform to any other cells. It can become heart cell, it can become skin cell, it can become reproductive cell, it can become a number of cells. So that is the stem cells are undifferentiated cells with capacity to both differentiate and multiplied into 200 cell types that form in human body. Can take any form. That base cell is called stem cell. You can collect stem cells from all these. You can collect stem cells from kidney, sperm, blood, all this. But for application, you need it in certain quantity, so that can only be collected at specific times. Now, this is a very promising technology. It has huge amount of application. You learn how to XYZ apply this stem cell technology. You can generate organs. You can generate all these diseases. So many brain diseases, Parkinson's disease, Alzheimer's disease. If you know how to regenerate the brain, then all these diseases can have some solutions. Now Alzheimer's absolutely don't have any solutions. You are left that people to either manage it till they die. Otherwise there is no absolute treatment for many of these diseases that is written here. Okay, organ transplantation, all this, it is so much helpful because so many times what happened, the exact specification of the organ is not possible. If you know how to handle the stem cells, you can produce exact organ what is required or adaptable to the XYZ body and all. Go ahead. What is transcriptome? We have seen genome. What was genome? Total DNA of all these organisms. Transcriptome is total RNA of all these organisms. Okay. Then there is something like proteome. What it is? This is first it will be DNA double helix. Then this is the same DNA that is getting transformed into RNA by splitting into single helix structure. Now it is this RNA that is going to the XYZ organ and getting transformed into proteins. And this collection of proteins are called proteome. Now having control of all of this, that you call as biochemistry of the cell, involving all whatever compound is formed, coming in general, going all this, having complete information, you call it as biochemistry of XYZ cell. At least know to these three terms. Go ahead. Genome is DNA, this is RNA, this is proteins. Three different stages in whatever the function they are doing. What is CRISPR-Cas9? I'm not spending too much time on this. One is too much common technology. Everywhere it is there, and UPSC also 2 times asked this. What is CRISPR-Cas9? It is a gene editing technology. What it does, there will be a target sequence, it go and Cas9 is a protein, will go and cut this. Then whatever new element you have to add, it will be added into this. Then you will get a new structure to the, it has made this gene editing technology too much easy. So that's why gene editing you see today in every applications. Because earlier it was too sophisticated technology. After CRISPR-Cas9 came, it becomes extremely easy process. Even colleges, universities can have CRISPR-Cas9 labs and all. And with including it with improvement in machine learning. So that now you are having software, you simply design into software, you give it, you get this edited gene. And try your plants, try your XYZ animals and all this. Almost all gene editing you see is based on CRISPR-Cas9 only. Even if it is corona, even if this technology was applied for, is it applied in humans? There was a case of a Chinese doctor using this gene editing babies. He created gene edited babies. He also was using the same technology only, CRISPR-Cas9 technology. Now rest of the detail, how it works, what is CRISPR-Cas9 exactly, these all you can read from here. These are different steps in CRISPR-Cas9 processing. Go ahead. What is Human Genome Project? Decode the entire base structure of human genome. One cell, like we are saying that thymine bases we were speaking about. So getting this entire information, is Human Genome Project. This is a global project. It was having all these 35,000 genes, this much of all this were done. When this project was going on, started, this is an old project, 1990s it started, at 2002, 2003. During this time, the results were published. Why we are discussing this now? 2 reasons why we are discussing this now. One is, even though it was said that human genome is decoded, we already have this information of human genome. Even though it was said like that, it was not possible for doing 100% human genome data. Only 92% of human genome data was decoded at that time. 8% was not decoded. Why? Because they are like satellite, they are not doing any key functions and they are too huge. So get this information was quite difficult and their output is almost negligible. So due to these reasons, we haven't decoded the 8%, pending 8% in the Human Genome Project. Now what happened? In recent times, this 8% is called repetitive DNA. Now in recent times, in 2021-22, this 8% was also decoded. So now it is saying that complete human genome is decoded. Now what next? Once you get complete human genome data, what can be next? This research is having 2 dimensions. That is application part that we will come to. What is first part? This human genome is done for a very limited sample space. Now you are doing this for India is having its own genome project, different races, different type of people. You are trying to get their genome data also. So that's why India is having India's own genome project. Other countries, other races are having their own genome project. And what is the next major advancement to this? Once this whole genome project is about getting the data. Now the next level is genome printing. What is it? You know where this cell is, you know all the information, you do something and make this, and try if you can make a human, or try if you can make a live DNA, active brain cell, or whatever it is. That is the next advancement of human genome project. What else? This is all written here. Human population on different earth is different. So we are having different types of genome project. That's why India is having India Genome Project. This is why we discuss this human genome project. Otherwise, it is a decades-old project. India is having IndiGen program. What is it? Making a whole genome sequence of thousands of Indian individuals. It is done by Council of Scientific and Industrial, CSIR basically. Now there is a Genome India project. What is Genome India project? To aim to collect genetic sample of 10,000 Indians as a reference. This is done by DBT. Human Genome Project, we already discussed this. Roadmap to future, DBT is doing, but 22 other organizations are partnering. It's not like only DBT is doing that. Different Center of Excellence and other organizations are part of it. 10,000 human genome will be analyzed for this project. Go ahead. What is RNA interference technology? Gene slicing, silencing. Tell me, what is it? Yes. ठीक है, RNA interference pathway. This all information is given here. Not going too much into it. Basics of RNA you have learned. Now read this RNA interference. This is also one time UPSC is asked. Less chance again a question come, but still this was in news. That's why we kept it here. Not going to details. What is DNA vaccine? mRNA vaccine we have discussed too much. We have different vaccine types. What is mRNA vaccine? Tell me that very fast. Which were these mRNA vaccines? Which vaccines were mRNA vaccines for COVID-19? Pfizer and Moderna were these mRNA vaccines. Now tell me, what is the new technology that mRNA vaccine is using? What RNA does? RNA give instruction to create XYZ protein. What mRNA vaccine does? It give instruction to create. No, COVID is having these kind of spikes. So mRNA, this messenger RNA will give instruction to create these spikes. Not create COVID virus, just create this spike. Now this spike will be created if you take this vaccine. This mRNA will create these spikes. Now if these spikes come into your body, what will happen? WBC will come and attack this. Once WBC killed all these spikes, it will have its memory how to handle these spikes. Now when original virus come, the spikes, same spike will be there. Your body WBC can go and easily attack this. This was all about mRNA vaccine. DNA vaccine means you are again going one step ahead. Your RNA is converted into DNA. Then you are creating a complete DNA structure and introducing this DNA structure into the body of this XYZ patient. Which vaccine is based on DNA technology? There is no vaccine right now available based on this DNA technology. What is major advantages? Infectious diseases, you can create vaccine very fast. Like this platform, if matured even mRNA technology, it is much faster way to develop vaccine. Otherwise, earlier polio vaccine and took decades to make. Both human mediated refrigeration is not required. Extreme conditions for keeping this vaccine, all this can be eliminated. Most of the mRNA vaccine benefits are equally applicable in DNA vaccine also. बताओ. Yes, yes. That mRNA is derived from COVID-19 virus, but there will be some editing done on it. Not exact mRNA will not be used, but it has been taken from COVID-19 vaccine and all. First, you can go through this. No, currently no vaccine is based on DNA vaccine technology. These are like different vaccine approaches. Just firstly go through this. What is viral vector vaccine? Tell me. What it does? You can study from here. Give me examples of inactivated. What is mean by inactivated vaccine? Or killed vaccine? You take a virus, you kill it. Kill it means it will not reproduce itself so much so that it can affect your body. So you keep away its ability to multiply itself, then introduce that virus into your body. So the same process happened. WBC will come and kill that virus. WBC will learn how to kill. Now then when an original virus come, body will already be learned how to handle this. That is inactivated vaccine. What is it can be very easily produced because you just need to take the XYZ virus and cut it some parts and make it the vaccine. Now what is subunit vaccine? Instead of introducing the whole vaccine, only a small part of this vaccine like spikes or some other parts will be only be introduced into it. But this will be parts of the vaccine, parts of the virus itself. In a mRNA vaccine, this part is produced by your body. Here the parts will be introduced itself. Now what is this? What is other type of vaccine? Viral protein is there. mRNA and dRNA vaccines are there. These are new technologies, mRNA and dRNA. Viral vector vaccine, what is it? Virus itself will be introduced at your body at much smaller dosage so that it doesn't create so much issues in your body. What is major issue with using these viral vectors? Risk of reinfection is there. Polio, you can see that somebody get the vaccine and from the vaccine you can get infection. So this kind of cases come in case of viral vector vaccines. Go ahead. These are different types of vaccine. Just for information, it is kept here. That's all from biotech and healthcare sector. Mostly all these are very factual informations. You simply have to read this multiple times and get the concepts clear. That's all for now. Thank you.